Hello everyone, today we are going to see the next type of digestion, that is digestion of protein. First of all, we need to know what is that protein and in which food product the protein that is present. The source of protein basically is meat. Okay, so in meat, in the fish, in egg, in milk, in all those we have the protein. So this protein, the main chief component, suppose if it is the carbohydrate, the chief component of the carbohydrate is what? The polysaccharide, then the oligosaccharides and the monosaccharide. In case of the protein, see, it is present in meat, it is present also in fish, then egg, then milk. Okay, right? So here these are the sources. So in all those sources, we are going to have the protein. Now this protein that is present, this is a chain. Okay, suppose this is a chain of protein. So in that protein, there are two terminals present. So in one terminal, that will be a amino terminal, and another terminal that is a carboxylic acid terminal. Right? So it is present. This is the structure. What is the unit? See, this is the protein. Right? So this protein that is broken down into smaller polypeptides. Okay, suppose it is broken down into the polypeptides. So these are the polypeptides. Then the polypeptides will be eventually broken down into, this is a tripeptide, this is a dipeptide, and eventually this is the amino acid. Okay, what is the unit here? That is the amino acid. So here these are the polypeptides. Okay, this is the protein. So this protein is a complexed most component biomolecule, and this protein is made up of the unit, and that unit is amino acid. Now just see, whenever it comes to the digestion of protein, in how many regions the protein has been digested. See, first of all, whenever we take the food into the mouth, in the mouth basically the carbohydrate is digested. In the esophagus, same carbohydrate digestion will continue. Whenever it reaches the region of the stomach, in the region of the stomach, the protein digestion will start. So first of all, just see in the region of the stomach what happens. First of all, we have one enzyme that is released from the stomach wall, that is the pepsin. But whenever pepsin is released, pepsin is not released in its activated form, right? So first of all, the chief cell, whenever it releases the pepsin is released actually in pepsinogen state. This is the inactivated state. At the same time, what happened? The other cell I told, that is the oxyntic cell, or that oxyntic cell actually will release the hydrochloric acid, HCl, right? So this HCl will convert the inactivated pepsinogen into the activated pepsin, right? So remember, it will convert into pepsin, then only this pepsin will work on the protein, and it will break down the peptide bond, and it will convert into small polypeptides. So peptones, it is nothing but the polypeptide chain itself. Now you can see normally what happened to the protein. That protein that is present, suppose we have taken the meat, then it will be acted upon by the pepsin. Now the next one, we have also another type of protein that is present in the milk, that is called as the casein. So this casein will be acted upon by another enzyme that is released by the same stomach wall, that is renin. But same thing is happening, that the renin that is released in the pro-renin state. Okay, remember only one thing, that if it comes to renin digestion, that is helping in milk digestion, that will have double N. Remember, never write single N. That will a different hormone that will be needed in the excretory system. Here in this case, that is the renin, RENNIN, which is released by the stomach wall, that in the form of prorenin. So this prorenin, in presence of the pepsin, okay, in presence of the pepsin, right, it will be converted into it will be converted into the renin. Okay, so this renin, okay, this renin is actually released in the prorenin state in presence of the pepsin. So this pepsin will act on this prorenin, which will convert the prorenin into renin. Then this renin will act on the milk protein and it will convert into calcium para-caseinate. In presence of calcium, it will be converted into para-caseinate. So that will occur in the region of the stomach. So in the region of the stomach, what happened? Our protein that will be acted upon by the pepsin to convert into the peptones. Peptones are nothing but the polypeptides. Next one, the casein, which is the milk protein. In presence of renin, it will be converted into calcium para-caseinate. Now, after that, the food from the stomach, earlier it was a long chain of polypeptide. It will be broken down into small polypeptides, suppose. Okay. Then after that, it will go to the region of the duodenum. In the region of the duodenum, what happened? It means the pancreatic juice. So here, this pancreatic juice that will contain the 3 different types of enzymes. So what are the 3 different types of enzymes? They are trypsin, chymotrypsin and carboxypeptidase. Remember that trypsin, chymotrypsin and carboxypeptidase that is released from the pancreas. So here it is occurring in the region of the stomach. In region of the stomach, this digestion occurs. In the region of pancreas, that means in the region of duodenum, the pancreatic juice will release the 3 enzymes, that is trypsin, chymotrypsin and carboxypeptidase. But remember that if it comes to the pepsin, pepsin is released in an inactivated state, that is the pepsinogen. Okay, so that pepsinogen that will be acted upon by one enzyme, that is the enterokinase. Okay, and this enterokinase will convert the pepsin here this is the trypsinogen, which is the inactivated state. So this trypsinogen, in presence of the enterokinase, will be converted into activated trypsin. Okay, so this trypsin that will act on the protein, because 100% protein digestion will not occur in the region of the stomach, it will reach the region of the duodenum. So the protein that will be acted upon by now the activated trypsin to convert into peptones and the other polysaccharides. Now after that, this chymotrypsin, have you seen? So this chymotrypsin actually will be also released in an inactivated state, that is chymotrypsinogen. So this chymotrypsinogen will be acted upon by the trypsin to convert into the chymotrypsin, right? So this chymotrypsin will act on this protein, milk protein, to coagulate. So you can see that milk protein, earlier it was in the state of the calcium para-caseinate. So this calcium para-caseinate that will be acted upon by the chymotrypsin to convert into a coagulate state. After coagulated milk protein, it will be absorbed. So this chymotrypsin that will act on the milk protein and it will convert into the coagulated milk protein. Then the next one, that is the peptides that is left. Here you can see this polypeptides or the peptides. That peptides will be acted upon by the carboxypeptidase, which is also the another enzyme released from the pancreas in the region of the duodenum, act on the peptide to convert it into the dipeptides and amino acid. Dipeptide means it is going to have only 2 amino acid bind by a peptide bond. So it will hydrolyze the peptide bond and it will be broken down into the amino acid. So these 3 enzymes, that are trypsin, chymotrypsin and carboxypeptidase, that is released from the pancreas in the region of the duodenum, the protein will be converted into the polysaccharides or the polypeptides or in the state of peptides, right? Then after that, come to the next region, that is the jejunum and the ileum. So you can already understand that the jejunum and ileum, they are going to have the crypts of libercune or the crypts of libercune that will have the intestinal juice, and the intestinal juice that will act on the peptide. They are releasing actually two enzymes. One is erepsin and the other is dipeptidase. So erepsin will work on the peptide and it will convert into dipeptide and amino acid, and the same crypts of libercune which is releasing the enzyme, last enzyme, that is dipeptidase, which will work on the dipeptide which is attached to each other by means of one peptide bond. It will hydrolyze that peptide bond and it will convert into the amino acid, that is the unit of the protein. So in the region of the jejunum and ileum, okay, in the jejunum and the ileum, what happens? This dipeptide and dipeptides that will be acted upon by erepsin and dipeptidase, and eventually it will convert it into the amino acid. So this way the protein digestion occurs. Remember one thing, that the protein digestion will never occur in the region of the buccal cavity and in the region of the esophagus. That carbohydrate digestion will occur in the region of the buccal cavity, esophagus, then in the intestine. If it comes to the protein digestion will start on the region of the stomach by the enzyme, that is pepsin and renin, go to the region of the duodenum by the pancreatic enzyme, that is trypsin, chymotrypsin and carboxypeptidase, and it will be converted into the polypeptides, and it will go to the region of the jejunum and the ileum. It will be acted upon by erepsin and dipeptidase and convert into the unit state, that is the amino acid. So this way the digestion of protein occurs. Digestion of protein occurs in the stomach, in the duodenum, in the jejunum and in the ileum by the gastric juice that will have the pepsin and renin, by the pancreatic juice which will have trypsin, chymotrypsin and carboxypeptidase, and the intestinal juice that is released from the crypts of libercune, that is succus entericus, that will be dipeptidase and erepsin. So that's all about the digestion of protein. I hope you remember. You just remember this way so that in the chart you can understand everything and you can write by your own. So this way just make the notes how the digestion of carbohydrate, protein, fats and nucleic acid occur. So that's all about the video. In the next video we will see the digestion of the fatty acids and nucleic acid. So that's all about the video. Thank you.